Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Melanocyte. It is a specialized dendritic cell in the epidermis, particularly located in the stratum basal layer, which synthesizes and transfers melanin to the surrounding keratinocytes. It is actually the organelle melanosome which contains this melanin is being transferred, not directly the pigment melanin. Coming to embryology, melanocytes are derived from neural crest cells. They will reach the skin via two pathways. One is dorsolateral, another one is ventral pathway. To see in detail about embryology, you have to go and watch my full video on melanocyte. This melanocyte precursors reaches skin via two pathways at around 10 weeks of gestation. They will not only reach the skin, they will reach certain extracutaneous sites like eye, inner ear and leptomeninges. In skin, they are based on the location, they are called follicular melanocytes, which contains melanocyte stem cells in their bulge, interfollicular melanocytes and acral melanocytes. Total 4 types. One is follicular, second interfollicular, third acral, fourth extracutaneous melanocytes, wherever they may be. Coming to demonstration, on light microscopy with H and E staining, it will look like a clear cell which is located in basal layer. So basal, low level, clear cells with small oval nucleus. Organelles and dendrites we can't visualize properly under H and E staining in light microscopy. For that we have to do electron microscopy. Special stains like Mason Fontana stains melanin into black. Enzyme histochemistry like DOPA reaction positivity because of the enzyme tyrosinase which is present in melanocyte. Immunohistochemistry melanin or MART1 positive, HMB45 positive. It is an antibody which goes and binds to pre-melanosomal protein also known as GP100, which is the major structural protein of melanosome. S10, S100 and SOX10 positive. This is about immunohistochemistry of melanocyte. Coming to electron microscopy, we can see a dendritic cell with oval nucleus and melanosomes. This is the main identifying feature, that is a specialized organelle melanosome. These melanosomes are present in 4 stages. Stage 1, round pre-melanosome without any melanin, without any enzymatic activity. Next, stage 2. Here you can see a scaffold-like matrix composed of longitudinally arranged parallel fibrils, which is mainly made up of protein, pre-melanosomal protein, that is PMEL. Here you can see longitudinally arranged parallel fibrils. This is the structural framework upon which our melanin comes and gets deposited. In stage 3, you can see melanin coming and depositing on the scaffold-like structural framework. Now finally stage 4, which is a mature melanosome which contains full of melanin that obscures the matrix itself. Here you cannot see any longitudinal parallel fibrils. So it is the final stage of melanosome, stage 4. Mainly the stage 3 and stage 4 melanosomes will get transferred to the keratinocytes. Here you can see all the features which I have described. Stage 1, which is round, 2, 3 and 4 in oval shape. Melanin will be increasing with increasing stage. No melanin, minimal melanin, moderate melanin, finally highest amount of melanin. Enzymatic activity will be nil in stage 1, high in 2 and 3. Finally it will become low at stage 4. Coming to melanogenesis, which happens inside the melanosomes. What are the stimulators for this melanogenesis? Mainly UV radiation, melanocyte stimulating hormone and ACTH, which also has MSH-like activity. First step in melanogenesis is conversion of amino acid tyrosine to DOPA, then DOPA to dopaquinone. Both these steps are mediated by our rate limiting enzyme tyrosinase. Next, if cysteine is present, it will convert to cystenyl DOPA and it will form pheomelanin. If cysteine is not present, it will convert to dopachrome. Dopachrome gets converted to dihydroxyindole, DHI. If TRP2, tyrosine related protein 2, also known as dopachrome tautomerase, if present, we have to add extra 2 letters to DHI, that is C and A. DHI carboxylic acid. It forms DHI melanin, which is brown melanin. Here DHI forms DHI melanin, which is black melanin. Both these brown black melanins are eumelanin only. Here simple flowchart. Tyrosine to dopa, dopa to dopaquinone by same enzyme tyrosinase. Then if cysteine present, cysteine dopa to pheomelanin, simple yellow red melanin, which is present in fair skinned individuals. If cysteine is absent, it will form dopachrome, then DHI melanin, which is black melanin. If TRP2 and 1 are present, DHICA melanin, that is brown melanin. Finally, eumelanin is brown black in color, pheomelanin is yellow red in color. Eumelanin gives more protection from UV radiation and oxidative stress. So there will be less chance of skin cancers, obviously, whereas pheomelanin gives less protection from UV rays and oxidative stress. Hence, there will be more risk of skin cancers when fair-skinned individuals gets exposed to sun. Coming to epidermal melanin unit. Each melanocyte transfers melanosomes to nearly 36 keratinocytes. This one melanocyte is to 36 keratinocytes, together called as epidermal melanin unit. This number of melanocytes will be same in all individuals, but why each individual is different in color? Because of melanosomes size, number, distribution, their degradation rate, activity and type of melanin. These are the factors which differentiates the skin color between individual to individual. What is the density of melanocytes? It is 1000 to 2000 per mm square. Highest in face and genitalia, lowest in trunk and acral skin. Acral skin like palms and soles will have lowest amount of melanocytes. Coming to functions, as we have already discussed, it determines the color of the skin, hair and eyes. Those beautiful blue eyes certain celebrities will have like Aishwarya Rai are because of variation in this pigment melanin. Low melanin, blue eyes. Pigment production and its transfer to keratinocytes. Provides protection from UV rays and oxidative stress. These melanosomes which entered the keratinocyte will form a cap or umbrella above the nucleus, protecting this nuclear DNA from UV radiation. It also helps in vision, auditory and immune function. Coming to applied aspects, hyperpigmented lesions will be present in phthalates, also known as freckles. This E and P is for excessive production of melanin. In melasma also there will be excessive production of melanin. Quite opposite, in albinism there will be nil melanin. A for absent melanin. Melanocytes are normal because of tyrosinase gene defect, which will lead to depigmented skin lesion. Now coming to increase in number of melanocytes. Benign localized increase is seen in lentigens and melanocytic nevi. That is nothing but common moles. Malignant melanocytes are increased in melanoma. Quite opposite, decrease in melanocytes or complete destruction of melanocytes. Vitiligo. Next, final applied thing is nevus achromicus, absent color, depigmentosis. Here melanocytes are normal. Melanosomes are normal. Pigment production is also absolutely normal, but melanosome is not getting transferred to adjacent keratinocytes, leading to depigmented lesion. That is nothing but nevus depigmentosis. Clear? Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. До свидания.